Assalamualaikum. Assalamualaikum warahmatullahi taala wabarakatuh. Assalamualaikum. Tes suara. Tes 1 2 3. Tes mic 1 2 3. Tak ada keluar. Tes mic 1 2 3. Perhatian kepada semua pendengar dan penonton, kita akan bermula sebentar sahaja lagi.
everyone. Hello. Hello, hello. Good morning. Hello. Assalamualaikum. Good morning. Assalamualaikum. Good morning. Hello. Assalamualaikum and a very good morning. Yeah.
Testing. Testing, one, two, three. One, two, three. One, two, three. Testing, one, two, three, three, one, two. Test. Testing, one, two, three. One, two, three. Testing, one, two. One, two, three. 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 Testing, one, two, three. Percobaan satu, dua, tiga. One, two, three. Testing, one, two, three. One, two, three. One, two, three. Testing, one, two, three. Testing, one, two, three. One, two, three. Kalau tak tu tukar tukar je. One, two, three. Percobaan satu, dua, tiga. Cobaan satu, dua, tiga. One, two, three. Cobaan satu, dua, tiga.
Test mic 1 2 3. Test mic 1 2 3. Test mic 1 2. Test mic 1 2.
Dua-dua deh. Okay. One, two, three. One, two, three. Tadi cakap dengan takut jauh sikit hilang. Saya kena pegang, Pak.
Susah aku important.
Kalau jauh dia tak dengar.
Yeah, okay then. So do I turn? How do I turn on this one? Okay, so I turn off first.
Okay, Assalamualaikum, good morning, and a very warm welcome to all distinguished guests, respected colleagues, and dear students. It is my great pleasure to welcome you to ICA Visiting Lecture Series number 6 for the year of 2026, entitled Emerging Trends and Development in Biostimulants. Thank you for taking your time to join us for this meaningful session. We are truly honored by your presence, and we hope that today's program will be insightful, engaging, and beneficial to everyone. To begin our program, it is my honor to invite our respected Director of ICA, Professor Dr. Hisham, to deliver the welcoming address. May I respectfully invite Professor Dr. Hisham to the stage. Thank you.
Okay. بسم الله الرحمن الرحيم والصلاة والسلام على سيدنا محمد وعلى آله وصحبه أجمعين. Selamat Hari Raya. Once we are in Shawwal in Malaysia, the whole month is Selamat Hari Raya. It's our great pleasure and honor for having our honor guest today, Professor Joe. And thank you very much, Prof. Ida, for joining us, and Prof. Zita. She's the head of Campus Pago and deputy director of Campus Pago. And actually, I'm very thankful to Dr. Jabir for bringing us these linkages and convince finally Joe to come to see and visiting us. I know Joe since almost, I would like to say 7 to 8 years, almost like this. We are both fellow in BGPR International, and we share a lot of things, a lot of activities, a lot of discussions. But Dr. Jabir make it reality and bring him here for that. I hope you're going to enjoy. In a personal, I would like to say thank you. I would like to consider Dr. Joe as my twin. We born in the same year. I will not like to reveal the year, but he's 1 month older than me. And surprisingly, when we discussed, we share the exact number of H index, exact number of citation. And I was surprising about that during our discussion. But we are great pleasure and honor for having Joe with us today as a start for long-term relationship. I would like to thank you all and also our guests in the Facebook who are following us, Innovation Center of Agritech for Advanced Bioprocessing at Bago Campus, UTM Bago. And I hope you are going, not I hope, I'm sure you're going to enjoy the talk today of Joe. Thank you very much. Thank you, Dr. Rayhana. Thank you very much.
Thank you, Professor Dr. Hisham, for the inspiring and also warm welcoming address. Ladies and gentlemen, it is now my great privilege to introduce our distinguished speaker for today. We are honored to have Professor Dr. Jin Wang Hong Yang, or known as John, joining us today. So Professor John is currently a chair professor of sustainable cultivation at the Brightland Future of Farming Institute, Faculty of Science and Engineering, Maastricht University. He is a highly accomplished and multidisciplinary researcher with expertise spanning biological sciences, environmental engineering, horticulture, plant physiology, and sustainable agriculture systems. He obtained his PhD in plant science from Australian National University, specializing in hormone biochemistry and photosynthesis. Throughout his remarkable career, he has held academic and research positions across Sweden, Australia, the United States, and Singapore, reflecting his strong international presence and collaboration. Professor John has published over 200 scientific articles and is recognized globally, being listed among the top 2% scientists worldwide in 2024 and 2025. Very impressive. Okay. Prof. John focuses on sustainable food production, biostimulants and soil health, control environment agriculture, and nature-based solution for environment sustainability. So without further ado, please join me in giving warm welcome to our distinguished speaker, Professor Dr. Jean Wang Hong Yang. So this stage is yours, Professor.
So I'll use this one. Selamat Hari Raya to all my wonderful Malaysian colleagues. So firstly, I would like to thank UTM for this kind invitation to come to your campus to share something very close to my heart. This is actually quite an interesting area. So can I have the first slide? To continue my thanks, special thanks to Professor Dr. Hisham for the interactions over many years. And more importantly to Dr. Jabil who connected us. So I delivered a talk in 2022. Yeah. So that was a good introduction. All right. So this is the key area, and we have really done a lot of analysis, and UTM, especially this center, is at the heart of, I would say, Malaysian biotechnology leading to sustainable cultivation. So this is why I'm here. And for those of you, next time just call me Jean so that it's much easier. The key thing is this is the future for cultivation. And this is the reason why I've taken the trouble to come all the way to Malaysia. So the first part of my Malaysian visit is to help all the growers around Malaysia, and particularly not just the paddy, all the oil palm majors were there at Terengganu, at Kuala Terengganu for soils. So I've adjusted the talk, and the key thing is I want you to take note of the keywords like cytokinins. Air kelapa muda. It has to be muda. All right? The biochemistry will change. Your earthworms, your cacing, and this is a new model. We first proposed it much earlier, but it wasn't accepted until this year, where the main colleagues in European Union, Professor Dujardin, published in New Phytologist. So when you publish in New Phytologist, it's not only a tier one, it is a recognition that biostimulants have finally emerged in a very, very basic scientific journal. All right. So let me just go on so that let me see. Oh, it's not moving. Yeah. This one is working. Yeah. Cannot move. Oh, must point there, is it? Ah. So who am I? I'm your neighbor. So I'm the professor in Netherlands for 5 months, but I am a Scandinavian-based professor for the last 7 years. So I've just taken up the challenge of helping Maastricht University to set up the Future of Farming Institute. So this is a very important development in Europe. All right, and we bring you the latest technology. So you have to point in that direction. Okay, so I also adjusted this talk because I'm seeing the audience. So this is also a career advice. All right? The world has changed, and to be successful, you have to handle a few areas. All right? So all the young people here, this is like a career job fair. Okay? So you have to do a few areas. So I am a multidisciplinary scientist, trained as a biochemist, physiologist, but now I am actually doing a lot of what they call industrial processing, but at the start, not as extensive as what Professor Dr. Hisham is. So we are trying to collaborate and synergize. The keyword, synergize our effort using a multidisciplinary effort. So this is something that I would like to share, and I will run through this. So this is like career thing. So I am very deep for the hormones, for the biochemistry. But nowadays, and especially with the emergence of AI, the artificial intelligence, you need to know across what is happening from lab to right. That means you need to know from farm to fork. That means even if you are a technologist, you have to know what is happening in the ladang, what our farmers are struggling. Then you think about the whole value chain all the way to harvest and all the way down to food processing. And then the economics, the financial instruments, how to get grants, and all that sort of thing. So this is the model that I will encourage the young people to think. You have to be T-shaped. And it's also good when you exercise, so you become T-shaped for the men. All right? So this is a very important principle that my supervisors have given me that I would like to share. Quite far. Can you move forward for me? Yeah. So this is the Singapore connection so that everyone could really appreciate. So I'm one of the few, the bottom bit got cut off. I work in all the Science University of Singapore, National University of Singapore, Nanyang Technological University, and the new kid on the block. Earlier, in collaboration with MIT, the Singapore University of Technology and Design. So I'm one of the few that I shared with Dr. Hisham. I work in the entire ecosystem. And we are your neighbors. I'm your neighbor. Very near. All right? So we develop the entire industry, and we work with FRIM, we work with Malaysian Nature Society to take care of the orchids. But we also are able to commercialize. So this is the key gut book. And I'm very happy to announce to you that my first author, which is the former professor from Department of Biological Science, receiving the highest honor in horticultural science as of yesterday, Professor Hugh. All right? So he's my master supervisor. But then later on, we realized that there's something missing. We have to go back to basic research. So I left Singapore to go to Australia. So looking at all the young people here, after you establish yourself at UTM, if you can get a postdoc or an attachment to go to Japan, like what Dr. Jabil did, do that. Then your horizon will be open. So if I remain in orchid, I would not be able to deliver this lecture. It is not easy to go to Australia, go to Europe, go to Canada, go to America, but the experience will be good because you learn new skills. And it is very hard to find a person able to do orchid, biotechnology, and all the way to the conservation of orchids. So if you have a broad mindset and a T-shaped thinking, this is a good approach. And you can see everything finally comes together for all the development, for your own career as well as serving the society. So everyone, I want you to take note of my classification about basic research and applied research. So this is a basic research. And orchids, because of the past British R&D support, Singapore had a head start together with University of Malaya. But later on, this whole industry actually moved to Taiwan. Can you remember all the beautiful phalaenopsis, the moth orchid in the hotel, in weddings? The origins actually start from Singapore Botanic Gardens. But we ride on the shoulders of the previous British science and technology. So it's not real Singapore per se, but we add on and we use our local orchid. And then it moves to China, it moves to Taiwan, and now everyone, it moves to Vietnam. So what does it tell you? At the end of the day, as what Dr. Hisham has said, the finances are important. Can the money sustain the companies after you translate the research? So this is a good example of applied research developed in Singapore. And this continues to be the mainstay globally. So this is my master's, all right, in Singapore. It's not moving. So this is the basic research on which that you will then see how we develop this formulation to have what you call sustainable agriculture, sustainable cultivation. So these are done mainly at National University as well as Nanyang University, where we have the best chemistry departments in Asia. This is something I can boast a bit. Singapore chemistry is really good. And I wish that we have more collaborations, all right, in the near future. So even though I'm Maastricht professor, I have to also say we are neighbors. And you can just send the samples across the causeway. All right? So you need the thing. And what is the best thing Malaysia has? Air kelapa. And a lot of muda one. Okay? So whether is it the old ones and the young ones, it has a big effect on your biochemistry, which I will just show one pathway. And once you know your pathway, you can sit it in a fermenter to make your artificial coconut water to sell to the whole world. Dunia, coming from Pago. And you have the best person here, Professor Hisham. All right, so let me just continue with the story. So the objective of this seminar, so this is where you are. Most of you are at the chemical fertilizer level, where you only look at inorganics, N, P, and K. But this is the reality. There is a hidden component that our society, Professor Hisham and me, we are part of the Asian Plant Growth, all right, Society for Growth Promotion using microbiome, all right? So this is where I'm going to take you to the next phase. Hopefully at the end of the lecture, you can reach to this. So it takes many years to get people to, your thing is not working too well. Can you go back one step for me? Okay. Hold on. You move one step. No, forward. Yeah, just one more to get the animation. Oh, the animation went off. Can you go back one? So most of you are at this stage. At the end of the lecture, I want you to know that inorganics and organics are important. And for the purpose of this center, you should be the Malaysian champion, ASEAN champion, and later Asian champion, hopefully for the world, that you will be the people producing the biostimulants that we're going to talk about. And of course, young coconuts are one of the best source of this biostimulant that will totally revolutionize, including oil palm. You can make oil palm greener, truly greener, not talk only. Really, really greener. So you are there at the precipice of big innovation. Dapan? So this is proposed, and people, this is published now in New Phytologist. So this is where we were. We proposed this in 2012, but nobody believed us. I delivered the lecture in Italy, but everybody is skeptic about this new thing. So people, this is inorganics. This is what is known. So the contribution from us is the A. A refers to the active compounds. So I propose a theoretical thing, and hopefully with the marvelous technology and designs of containers, Professor Hisham and this whole group could deliver the ultimate production of defined organics. So right now, most organics are not properly defined. But with proper fermenters and all that sort of thing, you revolutionize the whole thing. So basically, for those of you who are thinking practical terms, every typical Malaysian oil palm, the urea, the use of urea, you can have it. And if you can do even better formulation, you can drop to just 30%. Remember, it cannot be zero. You still need the basic building structure. So Malaysia is there already. So now it's to scale up, and this is the center, not only for oil palm, but for rice, for all forms of cultivation. Okay, so let me give you the basic. I decided to add the basic because after the talk, a lot of people did not understand. So this is just to give everyone the basic. And you will be quite shocked. So what are we dealing with? What are biostimulants? So everyone, you look at this. Why did John bring up plants and animals? Think about it. So what are we doing? So animals have a nervous system. But plants do not have an electrically based nervous system. So how do the roots tell the leaf, tak cukup air, not enough water. The technician is lazy, never come and water me. They send chemical signals. Of course, the one that you know is called abscisic acid, called ABA, right? So you know about all this, but you never have a coherent understanding. So everyone look at this. So hormones are produced in many parts. And it confuses a lot of students because they are multifunctional. So let me tell you, the same cytokinin that you saw opens stomata. But do you know it is the cytokinin that makes your chloroplast for photosynthesis? And it is not urea. So most people think that it is urea that make the leaves green. No. So all the plantation managers of Malaysia, their face turn white when I show this slide. Okay? So your whole biostimulant, you're actually looking at the hormones and other metabolites. So I'll show you a little bit more. And it is again another. So this is where we are talking about the bottom and the top. The top has a lot of challenges, insects. So this is called biotic stress. No water, this is called abiotic. And there is a lot of signals gathering here, and they send it up to the top. So all your formulation, you are actually playing around the so-called nervous system of the plant. Whether is it a big oil palm or a pak choy, a sayo. So all your formulation, you need to have this. And I was surprised that 95% of my audience, not just in Kuala Terengganu, throughout Europe, do not have this basic concept. Is this new to you, everyone? So when you talk about organics, you are playing with the signals. So here is a bit small, but you will be given this PPT. You have auxin. How many of you have heard of auxin? What does auxin do? Plant hormone for? You see? But you wouldn't know. That's why the first few slides are important. You're playing around the signaling from the roots to the shoot, from the microorganism to the roots, then up to the shoot. That's why I have to give you this grounding. So So you know what you're doing when you add cacing. What is cacing doing? Cacing is making more chlorophyll. But because you make more chlorophyll, you do not need urea, so much urea. Only very little urea. So if you don't have the fundamentals, you cannot advance. That's why I say today, I told Professor, I extend a bit, 45 minutes. The other day, I did just 30 minutes. And then they say, let's do another encore, a second one. So this is where you must go back to basic. So how do you formulate? How do you tell Professor Hisham, can you do enzyme 1, enzyme 2? You need to know what is inside. So think of yourself going to the hospital. Ayo, saya sakit, I'm sick. Take a blood sample. Have you taken blood samples from plants? Dr. Jabil, you only measure growth, right? Number of leaves. How to go to big tier 1 journal? You must do more sophisticated things. So remember, air kelapa is the real reservoir of the interaction between the xylem, the red color one, and the blue color one. And air kelapa is your best because it's easy. You can buy. Or you can plug your neighbor's coconut. Or get a mongek, the monkey, to go up to collect. So people, could you see our level of chemistry? One single drop of a maize, a jagung plant, we can run LCMS. If you use HPLC, you fail. We are already at UPLC. We are still doing HPLC. For certain things, it's okay. So your analysis is at the level of satu droplet. And we make our own instruments like this. Before you put in the grapes, you have to press out the liquid so that when you formulate for the farmers, you know what you're formulating. So that is the precision that Australian science brings to the whole thing. And we received the Australian prize in 1999 for this root pressure chamber. So we innovate. We build new instruments to meet the real challenges. Okay? So this is where early on we have already developed the concept of biostimulants. Okay? So how many of you have seen this machine, the one for photosynthesis? You have seen, right? So these two gentlemen, my supervisors, are responsible for the prototype and the scientific equation in 1975. The equation came out in 1978. So Graham Farquhar, our main supervisor on the left, received a Kyoto Prize. So what is a Kyoto Prize? It's equivalent to the Nobel Prize for subjects not covered. So this is the integration of sensing and the root pressure chamber. So if you put a certain microorganism there, straight away you can take a blood sample. 1 day, 3 days, 5 days. And then that liquid, you formulate, you find the best species of bacteria or fungus, you ferment that product, that will be your best biostimulant, and your center is there already. But you do not know what you're going to do. So this is the precision, and that's why they are well recognized. They have another Nature paper, 2022. Okay, so they don't have many papers, but one paper revolutionized the whole thing. That's why Graham got the Saudi Arabia highest prize. Then after that, finally the Kyoto Prize. If there is a Nobel Prize for plant science, Graham Farquhar together with Dr. Chin Wong will be there. So all the machine that you see, you borrow photosynthesis, we are the pioneers. So especially Professor Graham, who is the brain, Dr. Wong is the one, the engineering to build the tools using infrared gas analyzer. So just to give you the thing. So you cannot work alone. Everything is there. And we need the microbiologists to introduce the inoculants. You muscle there, you measure root respiration, then you collect at the leaves. And then your fertilizer with that thing will be the most effective delivery. And Malaysia can reduce, I would believe, 50% of your current inorganic fertilizer that comes mainly from China and elsewhere. Okay, everybody, you got it? So think of yourself, ayo, saya sakit. Oh, go and take a blood, right? But you never do it for your plants, your pokok. You just tambah fertilizer, no use. You must diagnose. So this is where we come in to give you the real big change. Very slow. You have to look at me. So this is my relationship with Dr. Antonios, formerly LIPI, sekarang is called BRIM. So all this is happening around Indonesia, less so in Malaysia, because you have Dr. Antonios championing the use of biostimulant and biofertilizer. So instead of applying so much urea, now you can reduce it. I heard they can reduce it to 40% for nanas. And I would say, I'm very proud to say, the nanas in Sumatra Selatan is better than the one at Pekan Nanas. They are, this is not bluffing, they are number 2 dunia globally. Great giant pineapple. And thanks to the biofertilizer formulated by our friend, Dr. Antonios. All right? So I'm a Singaporean. You see, I like to go Indonesia. You can really do serious trials. All right? So over in Malaysia, this is UKM. The same cacing vermicompost is actually improving the restoration. So all of you here, don't just think agriculture. This center will deliver even more good from whole Malaysia, whole Southeast Asia. You help in restoration of all the degraded forests, even for bakau, for your mangrove. Okay, but now it's a bigger challenge to Dr. Hisham. You need to have salt-tolerant microorganisms produced in biofermenters. So then, where's your market? The whole Gulf region, the whole Pakistan, parts of Africa. So your market is not just for agriculture. Mining companies will pay top dollars for restoration. Okay, so that is your market. And I'm the crazy one. You cannot get students from Australia. You can only get students from Malaysia, Indonesia to work in Australian mines because it is very, very tough. So I managed to get a girl from Johor. Yes, she graduated from University of Western Australia. Girl, okay. She very tough. Panas that at night in Australia becomes very cold. So what is the secret ingredient? It is humic acid plus fermented seaweed. The fermented seaweed can come from Terengganu, can come from Sabah. This one is humic acid campur with fermented seaweed. But the fermented seaweed you have to characterize using UPLC, MSMS, so that you know which one you're getting. And everything regenerate. So that is the contribution from the Asian people to the restoration of all the Australian mines. Previously, they never get it up because they tambah superphosphate and the acidity was. The customization from your center is very important, but your center will do better if at least some of you is able to understand the situation in the agriculture setting, and this is something new for all of you, in the mining sector. The grants from mining sector is very big. So the directors are here. Think alternate one. Think of the bauxite at Pahang. The unclosed mines around Perak. Okay? This is your market. The sandy soil. And Professor Hisham and some of you, you ferment your air kelapa, muda, and the other various species. The oil palm, convert to biochar, and then you add on all the things. You have it. You have it. Okay? So this is why they invited me for soil. I'm not a real soil scientist. I am an accidental because I follow the money. Grant got money, we follow. Okay? I'm Singaporean. I'm very practical. All right? Oh, ada, boleh apply. Then we apply. We got a, by the way, the grant is not obtained by me. This grant is 110 million Australian dollars. Not by me, by Professor Kingsley Dixon. He's very good in writing grants. So I learned from him. And my grant, the largest one, together with Ukrainian, we got 5 million euro. That's about 30 million ringgit. So you learn, you step on the shoulders of big people. So to the directors here, not just agriculture or horticulture, all the mines, think of rare earth elements. What are the microbes to handle rare RRE? I tell you, Malaysia has potential, but you need to open your eyes to opportunities. All right? So this is a reminder that soil science is in trouble. I showed this, but I know some people were offended. They never care about the biologicals. And this is a center gear up. So the soil scientists never considered soil. They're starting to consider something soil enzyme. But nobody measured the phytohormones. And the librarian in Sweden laughed at me. For 20 years, nobody borrowed this textbook written by University of California soil microbiologists and biochemists. 20 years. So, Prof, I can send you the PDF. Okay? So, all your enzymes, everything is inside there, but nobody borrowed. So the librarian was laughing. Itu gila orang Singapura come to Scandinavia borrow this book. Okay, then it turned out to be a gold mine. So this is what I'm recommending for our society and also for the whole Indonesia. All right? So, on your left, and then we move to the right. Everyone, the young people, are you ready for a career? You cannot just do your own area. Your basic science gives you good papers. This side may give you tier 2, maybe sometimes tier 3. You must have tier 1, tier 2, tier 3 for a complete career in your own development, and you feel happy. Because why? As what Dr. Hisham was saying, you feel satisfied when you can translate out your thing. So this is my career path. We are like a physics lab. It's physics. Then, of course, Gram and all that, their equation become part of LICOR. You heard of LICOR and all those things? It originates from Australia. All the basic science. Then you translate. And now, people, you could see, this is a mine in Australia. The microbes come from fermentation, and the machine, you see, is coming together. And you must not neglect tissue culture that I will show you later on. How? The young people, open up your minds a bit. So you became a T-shaped scientist. So try to connect the different disciplines as I walk through the definition. So let me now just to brush up your science. So how do you define a biostimulant? It is the microorganism and their products. But this is the key part, people. And this will be Malaysia's savior now that fertilizer cost has gone up. People, this is very important. You can produce the same oil palm, the same rice. You do not need to increase the chemical fertilizer. You have to find the right microorganism, all the products from fermentation. And you save all the farmers the money to buy the expensive fertilizer. Now, with the ongoing conflict, it gets worse. And this center should be ready to produce the liquid in bottles. Dr. Antonios have all the bottles waiting to be diluted, to be spread in the field. This center could do it. This is the scientific definition. Let's take a look at the government. All right. So this is the proof. I'll next show you the thing. People look. This is Chaching meta-analysis, and this is by Wageningen. It's at the bottom. You could not see. Everybody look. Every time you have earthworm, everything goes up. This is error bar. But what is the key thing that never changed? Everybody must look at this. That's all that you need. no change in the nitrogen. So it doesn't make sense. Your yield increase, your leaf nitrogen, your shoot nitrogen never change. So your biostimulant, when done correctly, is like a catalyst. And your center can deliver all this customized. Oil pump formulation 1, rubber formulation 2, Sayo formulation 3, kacang formulation 4, bakau a bit more expensive, saline formulation. You are ready to go for every Malaysian project. Then you expand further. So this is the European definition. I have to read up because this is legal. It is a product stimulate process independently of the nutrient content. People, the companies need this because they have to apply for license. So this is for the licensing part that you will come to it. All right? So all these slides you will have, but I'm just highlighting the key parts. Let's take a look at the function. So how do you tell the farmers all the complex chemistry? So basically, you have 100%. You tell the farmers, you are supposed to get 100% from your investment. But because you may be diseased, or you're unlucky, you get a disease. All right? So basically, your biostimulant close the gap. Easy definition, you're supposed to get 100%, but you never get it. So you apply this. And with your centers, mass production, it can be very cheap, and you get back your 100% or even more. That is the farmer's definition of biostimulant. Okay? Muhammad Sani, a fantastic PhD student. So this is that 20.2 impact factor. Our Europe most important environmental chemistry journal. These are the functions. And here you can see, you can have solid digestate that you can go on to make your biochar, or you use black soldier fly larvae as an animal feed. This gives the whole blueprint for Europe. And I believe this is also relevant for Malaysia. So you will have all these people, and when you click on Sunny and all that, it brings you to the journal. And all our things are open access. So people, that is the key thing. Your biogas plants could be running on oil palm fronds, of the bunch. You just get your methane, and after that, the leftover, you can either do biochar or the highest grade of biochar, nano biochar. And then you just coat it with chacheng liquid. You get the best fertilizer ready for Malaysia, ready for commercialization. Okay? So in a nutshell, please see biostimulants as a form of catalyst. And this center should be selling the natural catalyst coming from circular economy. And your name will be remembered throughout Malaysia for being the ultimate sustainable institute. So this is where we are right now for the thing. So just give you a quick overview. I will not go through the biochemistry. The current situation is that biostimulants have been not properly characterized. The chemistry is not there because the industry do not have sufficient funding. So a lot of failed results is because of poor chemistry. Okay? So let me just organize your thoughts into 8 categories, and you can see the overlapping. Like for example, if you use a bacteria, the bacteria may be operating through phytohormones and proteins. So this is very important. That means currently a lot of your natural products have overlapping. Like for example, the humic acid that everybody talked about, it is actually auxin. In the humic acid, our paper is coming out. So black soldier fly, have you heard of black soldier fly, the frass? Do you know what is the active ingredient? It's auxin and cytokinin, the chitin will break down to strigolactones and your salicylic acid that is giving the pesticidal properties. So your chemistry is very important. Okay, so people, it goes back to basic. So you can read this further, but let me just run through so that you get the full picture. So all is in this book and is open access. This is Belgium book, and the Belgian professor is the first author for the new phytologist article. Okay, so I'll leave this here. I will not go through. Remember, Dr. Young have to give you the legal framework. Just in case you're from the industry, and you'll be very grateful. At least you know where to go next. The legal framework, European acceptable definition. All right. So again, from that big paper, there are many products depending on your origin of your materials. And you will get different ratio. Like certain materials, you get more chitin from black soldier fly. If you get the seaweed from Terengganu, from Sabah, you get betaines, which is a very protective carbohydrate. So remember, it is not just the phytohormones, you also have a lot of other interesting metabolites. So hopefully your center has a metabolite platform, a metabolomics platform. Other. So you know I'm championing this. So unfortunately, my supervisor, he passed away over Christmas. So this is the man that is the father of modern micropropagation. So when you talk about air kelapa muda, this is Professor Leatham. He is the discoverer of cytokinin. So finally you saw who is the one who discovered cytokinin. It's him in 1962. I was his very last PhD student. Okay? So, people, this is santan. All right? All tissue culture, just take whole China, whole Taiwan, whole Thailand, and now Vietnam. Vietnam is taking over Chinese thing because China, the cost has gone up. Everybody, air kelapa, and for tissue culture, it has to be muda. But for santan, you no need to be muda. So let me show you a real operational factory. We have done all the LCMS, all the things. So I only want to bring you to this attention. I will not have time. This is the phytohormone complex. The phytohormone has to be protected from all the enzymatic things. So the formulation is there. So you cannot apply nakedly the hormone. It has to be a form of chelation, like EDTA and all that. So that, if you can characterize that, that could be a big paper in coconut. So that's the only thing I'm saying. So this is something, I added this in. I was in Vietnam. This is part of the Dutch delegation. Everybody in Vietnam, they pour away all the old coconut water because their economic value is the santan. You know, the kelapa to make curry. They only are interested in this. All this water, imagine you ferment it. It will be the super tongkat ali for plants, not for men. See, I know how to make Malaysian jokes. It could be a Kacik Fatima and all that. So I know my botany. So people, if Malaysian company is pouring away all this liquid, you're pouring away money. You go to Sigma catalog, you see the price of zeaxanthin, dihydrozeaxanthin, it costs more than gold. And in Vietnam, because they don't understand the biochemistry, and they are just chasing the santan market, could you see you have all that? I do not know about oil palm, but because they're in the same monocot family, they'll have it. I've already invited Sarawak to do the same for their sago. And sago is full of cytokinins, and they throw away or they burn it. So before burning, they should extract out all the expensive phytohormones that can be sold as the liquid for hydroponics aeroponics, and all the nutrient supplements. So this will give you an idea. Sayang, pour away, because we take this to sell as a santan for making curry. Okay, your center is ready, and remember, every time you do a fermentation, you valorize even more. Okay, I hope the idea sits in you now. So the next very important, cacing, and this is where we will still have to contribute to Malaysian, oil palm Malaysian. So they have isolated all the bacteria from the earthworms. This is done by Denmark. These species are known. You have to find the right strain to do your bioreactors. And of course, we are the first one. So Singapore cannot do a lot, but Singapore always have to be. I'm the first one to report phytohormone. Yay, promote. That sort of thing. So I got promotion. And this is NUS chemistry. Wow, bagus. So the key thing, you know that earthworms are good, right? But nobody show the mechanism. So we are a small group, not big grants. We show that the earthworm adapt auxin, to our surprise, the precursor for cell division. So Malaysia, don't wait. Set up your cacing farm, ferment it one step, and you have the tongkat ali for the whole cultivation. Oil palm, I can tell you, half or even more than half, they reduce their urea. Don't wait. This is too big an opportunity. Your center is ready. Your center is ready. So this one I have to just give you a little bit. Japan, they used to be the top. It used to be my boss. He passed away. And then it passed to Japan. And now I have to tell all of you, China has overtaken for cytokinin biochemistry. You know, China have to feed the people. There is not enough fertilizer, there is not enough farmland. You have to increase the yield. And increasing yield is not tambah more chemical fertilizer. You need the biological catalyst from Malaysia. Can be from coconut, can be from fermented cacing. All right? So what are we talking about? They already have this. So what we got from cacing is actually this too. Air kelapa muda gives you this. And Prof. Hisham, you look at this. This is easy for you. Beta-glucosidase. Sorry, I hope you don't mind. I campur the chemistry, then I give you the pathway also. I hope you like it this way. Because a lot of people, when they see this, they get headache. And you can take them from plants or bacteria. It's cheaper to get from the microbial pathway to come down. And people, this is important. Without this, the cell cannot grow. Nothing to do with MPK. All right, so let me give you the mechanism. All right, it is not just cytokinin, ada gibberellin and ada Bacillus steroid. So this is from South Africa. So we are combining all the data. So, Prof, this is for you. We didn't publish this. This is for you, and this is for this center. And who is our main supplier? Because we have a lot of unpublished data. This, CSIRO. People, different bacteria, different fungus like yeast, different strain. All your cytokinin. Remember, the Z is the one for plants to grow. Everything, whole dunia. Kada Z cannot grow. But the key thing is to find the nearest way to get to Z. So Z is the eukaryotic control, the catalyst. And you can do better than us. We are lab scale. You are industrial scale. And you can sell, this is more expensive than gold. So you can sell crude extract or purified using what they call HPLC. The time then you do your upscale HPLC. Okay? All right? Using C18 or C8 column reverse phase. So I think you know all this. So this is my suggestion to all of you. This is taken from Dr. Antonio's lab. He's on the right track. Prof, Dr. Antonio don't have a very good mass spec. That's the, that's the reason. All right. So for Malaysia oil palm, Malaysia has so much biomass. The best way is to use the biogas, and from there, you circulate all your things. In the process, in your biorefineries, you get biostimulant. You see, you have so many pathways this center can explore. And all these are published by Mohammad Sani, getting his PhD soon by Christmas time. Okay. So just a quick run through so that you don't miss out on the real thing. So I've only talked about the key phytohormones. The one that you may not be familiar, carotene. The rest I will not go through. So these are new groups for you to think about that you can also produce in your fermenters, and there is a big international demand. If you are a pest company, you will be focusing on jasmonic, stringolactone, and salicylic. This is the best for drought prevention, brassinosteroids together with cytokinin. This is for the entire hybrid rice program that Brunei and China is using, coming from fermentation of fungus. Okay, so I tell you, once you know this, you are in the controlling thing. But let me just show you the key difference between chemical fertilizers. So people, this is important. You can read this on your own. Fertilizers are running at milligrams. So a lot of people do not know the differences. Why do you call biostimulant a catalyst? What comes to your mind? Sikit, right? And it is in the nanogram. And because it's nanogram, this center can export easily in nanograms. So fertilizer, you're dealing with milligrams. And that is also to tell you, you need to invest in UPLC. You have to keep some HPLC running, but slowly, some of the analysis, you have to move up to UPLC. All right, so sorry to be technical, I'm a very technical person. Okay, and because it's nanogram, microgram to nanogram, that's why it's a catalyst. The catalyst control the milligram fertilizers. Could you see the understanding now? It is very powerful, and this center should do high value ingredients so that you meet Malaysian national goals of sustainable things. Okay, so just to keep this slide so that we go on to the real control. So the Z is very important. Without the Z, you cannot produce two. So this is where Australia always brings us back to fundamental science. So a lot of you didn't know when you apply biostimulant, what it does. It controls cell division at the G1S. Could you see the differences in education now, people? Dr. Young talking about tanam agronomy, then sekali go to mitosis. This is called the T-shaped multidisciplinary education that is missing right now. Because the real world, all the factors come together, but as humans, because of administration, soil science, industrial engineering. Because you partition, all these are lost. When you do your fermented liquids, you are controlling the cell division. So I leave this here, you read it on your own. Where the auxin come in, where the zeatin to produce two cells. So all the oil palm colleagues had dinner with me. Dr. Young, we are wrong. But we don't have the money. Malaysia, MPOB, not funding fundamental research. But I say you don't have to do all. Just do a little bit. I suggest to them to come to UTM to try their formulation. But if you can do a cell assay to check whether more cell division, you will get a lot of money from MPOB side. Okay? So just give you one example and a molecular control. Control, remember I'm talking about Cine fungus, sending up gibberellins. And for like that Chinese student over there, you didn't even know this is a Chinese name for gibberellins, right? That's why I say, habis lah education. We need to really relook at the education. So people, this is a controlling. We are just looking at one, just one. Gibberellins alone here got 220. 220 types of gibberellin. Let's look at the big paper that China produced. It's unfair, but because they have big budget. For the first time, and people, it's published in Science, when you apply fertilizer, the regulator is gibberellin from non-tillering. So this will be similar for wheat, for oats, for all the cereals. This is in Science. So to use your nitrogen, you need the gibberellin. And in Malaysia, the gibberellin for the rice comes from your mycorrhiza. So you can either produce mycorrhiza for inoculants, or you give the farmers a fermented liquid with certain group of gibberellins before they apply the fertilizer. So that Malaysian, instead of 1 or 2 tillers, they get many tillers and the productivity goes up. And this is in Science. But of course, this group, I know them, they have big budget. How the world has changed. China has a lot of research money. That's why I'm going there next week. Okay, to Tongpang a bit, because I don't have such a big budget. Okay, so people, you look now with molecular, then they can solve, itu masuk, then it helps the fertilizer to go in. So this is the catalytic function of your biostimulant. This is real molecular data. Are all of you surprised? So a lot of people say, apply mycorrhiza, but what does it do? Nobody has the evidence. So thanks to molecular genetics, people are able to do this. So Prof. Hisham, finally, the European accepted our definition after 15 years. Hormones. They didn't accept when I gave the same talk 2012. So now it's a new phytologist. He is the champion globally for biostimulants. Hormones are important, and Malaysia has the best, together with Indonesia, have the best sources of things. So people, when you have my PPT, click on it, download it yourself. I will not go through the details. And the last group is proteins. The waste proteins is already in 2008, but a lot of people did not believe the results. Let me just give you the name of the main scientist. You can Google him and download the different proteins, especially from waste tofu. So people, this is the name, Professor Kola. He was the examiner for our student from Nigeria. So 3 big source, air kelapa, your cacing, and any proteinaceous waste, your enzyme, your mushroom. Remember the mushroom? Spent mushroom waste or the other parts of the mushroom. They will have all this, and they will have other nutraceutical. Mushroom will be different. Protein done, nutraceutical. So you need a very good LC-MS system to do that. So I will not touch on this because the mechanism is not clear, but likely to be through transcription factors. So my last piece of evidence to convince you and to close off the session. So to end off, let me just give you the last evidence. Remove the microorganism. And Japan has the best formulation. And you see the Japanese formulation, then you will be very surprised. This is Hyponex, the most famous, everybody follow. It is even better than Murashigi and Scoot, your MS medium. Everybody look at the Japanese. Adenine is the precursor for cytokinin. Japan keep a lot of secrets. You look, Japan already knew about peptones. Think of the PNAS paper, microprotein. Look, Japan buy a lot of air kelapa from Philippines. but they didn't ferment it. So Malaysia ferment it, you get more. So people, when you look at Japanese formulation, this is the best, and they don't publish. Japan already have it for years, but Dr. Young has to be the scientist to be open access to explain to all of you. So by looking at the tissue culture system, you know that you need your biostimulant in a medium without microorganisms. Japan knows it many years ago. And look at Singapore. Singapore is supported by none other than air kelapa muda for the entire, not just orchid, all the bunga-bunga, whatever bunga in our botanic garden, in all the factories around the world making tissue culture. Air kelapa has to be muda for tissue culture. You look, how do Singapore do the breeding? This one kawin, this one, a royalty come to Singapore. This one kawin, this one, another royalty. Then we do the diplomatic thing. That is how Singapore do the breeding. The pollen are all viable. And what is the nutrient? Air kelapa muda. Okay. So this is smart use of biotechnology. So back to Sumatra. So people, this is the important thing. So in tissue culture, no microbes. When you go out, they need a microbe. This is why every plantation has to have an inoculation program, whether it comes from your product or from compost, applied correctly. And then for the directors here, don't forget you have a new market, restoration. Okay, cannot just plant. You need to tambah back the microorganism or the fermented liquid that you have characterized to stimulate the soil health. So this is done in Australia. So you need to bring back the natural soil. So this is the key thing that you must always know. Who provides the biostimulant after tissue culture and also after seed germination? That is your basis of your center to do the thing. You need to convince the politician. Don't just check up soil health, soil health. You have to say what is happening. So basically, you need a catalytic function that your center produce at low cost, and that will reduce Malaysian costs in importing fertilizer. That means you become more efficient with lesser fertilizer. All right. So I will just add off here. This is a mechanism we have proposed, Prof. Hisham. Nobody except they laugh at us. So now they have accepted. So people, the key thing is just this, and I'll leave you with this slide. We are at, I'll come here so that you can better understand. We are actually here. Tambah the fertilizer, it grows. We have neglected this. So this is where everybody talk about soil health, but nobody has real empirical evidence. So people, your center, if you do this, you will really increase the yield, but also not polluting Malaysian environment. So this is what we do. This is your product. And Prof. Hisham, you can share this in your other talks also. This is actually the biosimulation that Prof. Reddy wants me to share, but I don't have time. So we are doing this. This is the real thing. And you change this according to your soil type. Whether you use a trichoderma, a fungal approach, or the plant growth promoting rhizobacteria. Thank you very much. Exactly 55 minutes, slightly over by 10 minutes. Can you just move to the last one? The last one. So this is the new definition.
thank you, Professor John, for that very insightful and also inspiring talk. From biochemistry to plant physiology and soil scientist by accident. Betul tak, Prof? Soil scientist as apa? By accident. Okay, ladies and gentlemen, we will now open the floor for short question and answer session. So if you have any question, feel free to raise your hand and kindly introduce yourself before asking the question. Anyone? Okay. Uh, okay, Professor John, I have one question actually. So, how can this biostimulant can be effectively applied to the plant? Is it by root, by the soil application, or by foliar application, where through the leaf stomata?
Okay, this is an interesting question, path of application. So I have to divide to two groups. If you remember at the start, I put down abiotic and biotic. So let me answer your second part of your question. You say through stomata. So the stomata refers to biocontrol. So for biocontrol, it's best to be applied as a foliar spray to prevent diseases. So for example, black soldier fly, the frass, you extract out the chitin. Of course, you know from the chitin, this part you wouldn't know. The chitin, that part will also have jasmonic and the most important ingredient, salicylic acid. That one you have to apply as a foliar, and a lot of diseases will be suppressed. And a lot of you will check in literature to say, hey, Dr. Young, how come salicylic acid synthetic forms when applied, it has no effect? So this is why the whole biostimulant requires synergistic. When I say salicylic acid, it is not just salicylic acid. It has like the sort of chelating agent. You go back to the I-kalapa, it's not only the zeatin. The zeatin is protected by a few other proteins that we did not characterize. Remember, we are running a LCMS, only getting the zeatin molecular mass is 219. We only get a 219. But the young lion, all the little microprotein, we never. So the salicylic acid isolated from black soldier fly, it will have some other components that I don't know. So for the industry, they treat it as a gray box, not a fully black box. So that you apply the natural one because it has the protection. That means it is salicylic acid protected by some other compounds that we have not. This one you need proteomics, and proteomics not cheap. Okay, so I'm also being very mindful of cost. So I'd rather have the natural product partially characterized. And then back to the root application, root will be cytokinin, auxin. Those are normally for your growth. And those are best applied to the roots. So hopefully that answers your question. So biotic factor normally at the top, soil drenching at the bottom for abiotic, fertilizer, lack of water, and all those.
Facebook, yeah. Can Secretary check, is there any question from Facebook? So far, no question. So far, no question. So again, is there any question from the floor? Any additional question? Yeah. Can I hand over the mic to you? It's okay.
Good day, Prof. You mentioned over and over again that we have a lot of resources, and we do agree we have a lot of resources. Could you please brief slightly how you convince the Australian government or the Australian funders convince and justify the ROI to get all the funds and how we could implement that with our potential funders and government agencies to do the same. Thank you, Prof.
All right. This I have to go back to the slide. Can you go back to the mining slide quite far back? Yeah. Okay, sini. So this is not my credit. Remember, we have a picture of Professor Kingsley Dixon. He was the former director of the King's Park at Perth, the very nice botanic garden. So people look at this. So many restoration projects did not work because the normal restoration project means to tutup. Once your mining finished, they just plant a few trees and disappear. Fortunately, all the trees died. So the government were under pressure. So how could the trees mati? So you look at this, people, everybody look at this. What happens during mining? Habis, no topsoil. The subsoil becomes, so you totally have a different system there. So basically, the subsoil have a different group of microbiology. So people publish and all that. And the government were under pressure from the native groups, the indigenous group, because everything mati. And what was their rubrics? So people think of the rubrics. They measure the NPK. NPK bagus, bagus, bagus. But none of them do the microbiome thing. No, it doesn't exist in their concept. Still traditional. So everything, you look at your soil test. Do you do microbiome or even the 20-year-old neglected book, hormones? So the government were at a difficult time. NPK of subsoil now at the top is very high, but not accessible. So there's difference between phosphorus being available and not available. So total P high, but the plant cannot use it. So they spend money, they plant the tree, die, plant the tree, die. All the contractors all get stressed. I get a phone call, 2015. I got inspiration from Kuantan. I went to the seminar at UNIZA. They're also struggling. So I got inspiration. I told my wife I need a break from the office work. So I went to Perth. Slightly less than 2 years. We fixed it using the concept from tissue culture, biostimulant. So you have to go back. So from here, 20 kilometers Adat reference site. What are the things? So it turned out to be Bacillus subtilis. Take Bacillus subtilis. We had a very good small contract. Tambah. Then cannot grow too well. Feed organic carbon to keep the bacillus as long as possible. And then we plant legumes, pigeon pea, rhizobium, and all that. So later on, we realized the best solution is to tambah humic acid fortified with correct microorganism and seaweed. The seaweed have the betaine to protect not only the plant, but also the microorganism. Because you tambah, the microorganism may not survive. So you see, it becomes interesting. So that is the process with the government. So finally the money comes, there is political pressure. It's big, that money for Professor Kingsley, 110 million Australian. Okay, so this is how you need to wait for the political thing, but you must get ready your science. So remember now all the restoration they are changing, improve soil health. That means they plant legumes, not so much for the nitrogen. The nitrogen is limited. The legumes is to increase the soil organic carbon, done soil phytohormones that will perpetuate the succession of the plants. Remember, you learn natural succession, but the natural succession never tell you actually the microbiome improves. So hopefully that answers your question. That is a big grant. That is a big grant. It's still running. Yeah.
Thank you for the question. Is there any other? question on the floor? Okay. So if, Prof, silakan.
Thank you very much, Professor. I think you're giving also a very nice insight. That's indicates not everything, as everyone say, available, non-available be, like example as people know. And also the role of microbiome, soil microbiomes impact how it's affecting that. But I think it's also another formula, which is very important, thinking about the micronutrients and minerals. Like in our body, when people say, I take potassium, 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 or sodiums or whatever, if there is no magnesium, it's never work. This means I have need to consider both. And also in terms of microbiome for their growth, we need also those micronutrients as a catalyst for the whole enzymatic system. How you balance this in your opinion for the soil rehabilitations? Have you faced this problem also about the micronutrients during rehabilitation?
Uh, the micronutrients comes from the very precise soil analysis. We actually harness two. You can't do, have to be technical here, ICP-OES. To really chase the micronutrient, we actually go down to ICP-MS. But for the smaller ones, ICP is not sensitive enough, you're down to AA. So you really have to map your reference site versus your current site that you buy, and you try to match it. So in the biostimulant formulation, normally we have a large amount of organic matter, that means your OM. So normally the best way, that means practical, is to put down precisely your missing microbe. Hopefully you get it right. And then onto established one like vermicompost or high biology compost. So there is a bit of black box in it. And together with the earlier reference, that means sometimes you have to tumble a bit like molybdenum onto your whole project on that sort of thing. So this is where site to site there are differences. But I definitely agree with you, potassium is big. Yeah, potassium is actually most of the addition. For certain site, magnesium is also important. So the whole thing is, it now becomes jumbo. Organic, but the organic becomes defined organics. And then precise inorganic supplement. So it becomes a joining together. So it's a real science-based analysis. Then you do the intervention. So it has to be done that way. It's not that anything you tumble, no. In Sumatra, we still very carefully check ICP. When ICP doesn't really work, you do the AA for certain things that you suspect. So it's that combination. But colleagues, can I tell you the best way that Malaysia have done? Malaysia plant the cover crop, mucuna. So all the plantation managers say, yeah, mucuna. I say your mucuna, the nitrogen fixation, BNF, biological, is very little. But what your cover crop is giving is your phytohormone in the rhizosphere. That is actually the missing part. So they got the concept, but the scientific basis is incorrect. So it was Graham, together with my supervisor who passed away, Professor Leatham, who showed that rhizobium not only give you the nitrogen, sikit-sikit, but also the full flux of phytohormones that now we call it biostimulants, the greater one. Prof, hopefully answers your question. Yeah, it have to be jumbo, jumbo, and charge the company money. Then you collect the revenue.
On top of that question, you do your do you do your chef life study, or I would say the sustainability, how long it takes, and then how many regulation that you need to do to ensure the phytohormones keep on flowing. Adding into the soil. Because I remember a long time ago, we do such things using the bacillus, but on the municipal waste length. However, the politician said it's not sustained, and they said normally they said natural things not sustainable, or they will mutate into something else. That's their worry. Thank you, Prof.
Now we are talking about persistence of your inoculum and over extended period. So, can you go back some more? Go back some more. Oh, sorry. All the way to the front. No, belakang. Go all the way to the back. I want very nice one to show you. Just jalan into my appendix. So the young people, you just go to the conclusion that side. Conclusion. The young people, not just prepare your slides, I must prepare things for additional question. Jalan, I'll tell you where to stop. Yeah. You'll be shocked. Dr. Young have a lot more that I didn't show you. Come, jalan. Stop. Belakang satu. Stop. Ta-da! So this comes from experience. Prof, this is the next grant application for 8 million euro. 8 million euro, 8 times 5. Okay, people, the young people, you must always think through your slides, anticipate the question. So why did the microbe did not survive? Okay? Of course, the first reason is it is what you call the population dynamics. The one that you apply may not survive simply because it ran out of substrate in the ground. So when you tambah your inoculant, it's best to have a bit of, I would recommend most of the time, you increase the organic carbon that they feed off. So in the media, what do you use as a source of media? Okay? So you may have some wheatgrass and all those things. So you have to think. So some people put in the inoculant together with partially composted material as a substrate to feed your organism of interest, so that it is the, what do you call, external one to be able to survive, while the local fellows will be pushing. There's competition. So that's how we do it. So this by the way, you have all these slides. I want you to, when you click on cantik, it goes in. But this is a scheme. So there will be all these metabolites. All right? So this you could see, you're talking about host adaptation. So you need to provide for your, like, bacillus will have a certain preference in your application onto your dose dump. I believe you never add anything to support your bacillus, right? I believe you did not. So this is the mistake I made also. So now the formulation, you have to pack the baby food for your inoculant, so to speak, so that they can persist. So this is the thing that we are thinking. But we also do two ways. Remember, when you apply microbe, the microbe doesn't work directly. The microbe works indirectly by producing the metabolites. So the best metabolite substitution is always your seaweed or your old air kelapa, the old air kelapa, so that it can contribute. That means before your microbial be able to do the de novo synthesis, your products are already there, and they are in your coconut. Remember, you can use all the hard part of the coconut. They always have fragments of some coconut flesh. That is the makan, or to keep your thing going, the food, microbial food. Or straight away you have sheep, seaweed, you cut them and you apply. So your inoculant done, this helper was sheep. So people, the schematics are very important. You will have this, all the people here, you have Dr. Yong's presentation, you push this part. All right, you push. And this is survive. Remember, the microbe itself doesn't give to the plant. They give indirectly through the metabolites. Remember, so if you don't have the right microbe, you can give the metabolite. Right? That's the coconut water. And remember, the coconut water is the xylem water, is the phloem water. That's why goes in very well. All right. So people, in future, this one will include zeaxanthin. Now you did the connection now. One of them could be the zeaxanthin, like some of the dot, I don't know which one, I can't remember, is the unknown. It could be a microprotein, it could be a transcription factor. I don't know. So this is your generation already for you to take over. I have to go and jalan-jalan already. Okay, it's your generation. Next time you say, oh, you add the zeaxanthin done to proteins. Wow, that is your UTM formulation. You patent that. Could you see the direction? So you have to feed your external inoculant when you go in so that it can persist. By the way, our strategy, it persists for at least 5 years. 5 years. So they go back to do the eDNA sampling to monitor.
Last question, maybe last question from me. Prof, when you mentioned about the biostimulants, and then we have new phytohormone, which is Carikins group, and so on. So, do you think these things can be in the textbook? Because I'm teaching plant physiology right now, and we don't have this in our plant growth related chapters. We stop until auxin, cytokinin, gibberellic acid, and so on. Please, Prof, I think the best thing is to have this. We need the textbook, Prof. Thank you, Professor Joe. So I think there are no more questions.
The slides are available. I give you all two versions. PowerPoint you can adapt for your own teaching, your own research. The PDF you can circulate to your friends because Maastricht University, we are totally open access, and Jabil knows we really have the funds to really do open access, including the lecture. So terima kasih for inviting me today.
Thank you, Prof. John, for the engaging question, and thank you once again for your valuable responses to all the questions that we asked just now. On behalf of the organizing committee, it is my great honor to express our sincere appreciation to our distinguished speaker, Professor Dr. Jin Wan Hong Young, for sharing his valuable knowledge with us today. As a token of appreciation, may I invite Professor Dr. Hisham, Director of ICA, to present a token of appreciation to our speaker. Please, Prof. 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 Ida also, please invited. Please be invited to the stage. And I think Dr. Zita, you can join, please. Thank you, Dr. Zita. Ah, sini. Uh, so please give round of applause to our speaker. Wow, UTM Mafla. Okay, everyone please stand for a group photo. Those yang dekat depan. Ha. Oh, ada lagi ya? Kejap. Alright. Okay. There is more one more things to give to you. Ada lagi bag. Sijil. Okay. Ada sijil. Another one certificate for the speaker. And also another one. Oh, another one small gift from me, ICA. Okay. Thank you, Prof. Isham. Thank you, Prof. John, Prof. Ida, and also Dr. Nozita. Ladies and gentlemen, with that, we have come to the end of today's program. We would like to extend our heartfelt thanks to our distinguished speaker, Professor Dr. John, our Director of ICA, Professor Dr. Hisham, the Director for the Head of UTM Campus, Professor Dr. Ida, and Dr. Nozita, and to all participants for your present and also active participation. We hope you have gained valuable knowledge and insight from today's session. Please do not forget to scan the QR code given for the attendance and also e-certificate purposes. With that, thank you once again. Assalamualaikum and have a pleasant day ahead.